well, let's go ahead and get started on these. So number one, genotype versus phenotype. Those often get confused. Genotype tells you the genetic makeup, so which alleles you have, right? So it's hard to talk about one of these terms without talking about the other one. These are genotypes, right? If you're homozygous or heterozygous, that's a genotype. Tells you which alleles you have. Phenotype is the trait that the alleles code for, right, or responsible for. So we think back to our Mendel example yesterday with flower color. An example of genotype would be like heterozygous, one dominant copy, one recessive copy, and the phenotype then would be purple, right? The purple flower color is the phenotype. So phenotype tells you which alleles you have. The phenotype are the traits that are determined because of that phenotype. So today we'll talk about Punnett's. We'll review Punnett squares and talk about genotypic ratios and phenotypic ratios. Okay. Homozygous versus heterozygous. Homo means same and hetero means different. So if you give examples of those, yeah, homozygous would either be homozygous dominant, two dominant alleles, or homozygous recessive, right, two recessive alleles. Heterozygous would be one of each, right? So hopefully that is pretty straightforward. And then lastly, dominant versus recessive. So generally when we talk about dominant versus recessive, we're either talking about the individual alleles or we're talking about the phenotype. So if we're talking alleles, the dominant allele is usually represented by the capital letter, and the recessive is the lowercase letter. Okay. If we're talking about phenotypes, the dominant phenotype is the one that shows up when you're heterozygous, right? The recessive one is hidden when you're heterozygous. So when we're talking about our flower color, the purple was dominant. So if you were heterozygous, as long as you have one copy of the dominant allele, you show the dominant trait. To show the recessive trait, you have to have both recessive alleles, right? So that's just kind of a quick overview again of what we went over yesterday. Is everybody feeling like 100% comfortable with these terms and no questions? Yes. Yeah, so I just put those in this morning. They're really good for the most part. I think like almost everybody in this class got a 10 or above. There was a couple exceptions, but I'm still waiting for one more person to take it. So try to get those back to you soon. Sorry it's taking so long to get them back to you. Okay, so I grabbed some notes out. I'm hoping this doesn't take too long to review. We're just going to do really simple monohybrid Punnett squares today. But we are going to throw in a couple other things with that you might not have gone into a lot of detail in at once. So I would still take notes. Definitely on paper out in front of you. Actually, unless you like have this PowerPoint pulled up, I really appreciate it if you put your computers away. If you're having trouble seeing and you have that pulled up, it's fine. But otherwise, I don't really want anybody looking at other class stuff. Is this 9.2? This is still 9.1.
How many laws do you have?
I think just two. At least two that we need to know. We're only going to talk about one of them today. And the other one we'll talk about when we get to like dihybrid traits. So, first law of heredity is called the law of segregation. So, probably more familiar with that term segregation in history, especially U.S. history context, because segregation means to keep separate or to separate. And so the idea behind this law is that during the process of meiosis, alleles are going to separate from each other and end up in different gametes. And so the alleles start to separate in meiosis 1 when our homologous chromosomes separate. And then in meiosis 2, the sister chromatids separate. So in the end, each gamete is going to get one copy of one allele only. And the chances of getting either allele from that parent is going to be 50-50. Right? If you're heterozygous, there's a 50% chance any gamete is going to have the dominant, 50% chance any gamete is going to have the recessive. I've got a couple of visuals to help you think about that. So here's our three possible genotypes, right? Homozygous dominant, homozygous recessive, heterozygous. These alleles would be on homologous chromosomes, right? And so when we do meiosis, they get separated from each other. So half of the gametes going to get one version, the other half of the gametes are going to get the other version. If you're homozygous dominant, even though they're getting separated, your gametes are going to get the dominant copy, right? That's all that's possible. Similar if you're homozygous recessive. If you're heterozygous, half of your offspring are going to get the dominant, half of your offspring are going to get the recessive. And again, if we take a closer look at meiosis, so this is kind of similar to that diagram from your quiz. We have a very simplified cell with one pair of homologous chromosomes. What happens to those chromosomes during meiosis 1? And then they separate. And then in meiosis 2, what happens? The sister chromatids separate. Okay. And so each gamete, each cell is going to end up with one copy of one allele. Okay. If you're heterozygous, half of them are going to have the dominant copy, half of them are going to end up getting the recessive copy, right? And this is the whole reason we can set up Punnett squares the way we do, is because there's a 50% chance, 50, that you're going to pass on either allele to your offspring. Here's just another look at that process in meiosis. So this segregation of alleles really happens during meiosis 1. And metaphase 1 is going to determine which gametes get which copy, right? So the paternal chromosomes on this side is going to end up in one cell, the maternal is going to end up in the other.
So metaphase 1 creates one.
Yeah. I mean, technically, technically it happens then. If crossing over happens, then sometimes we don't get separated until anaphase 2. But I think what's more important is that at the end, you end up with one sister chromatid in each cell, so every gamete has one copy of one allele. So half of the gametes are going to get one version, half of the gametes are going to get the other version. Feel pretty good at that concept? And I do think it's interesting. Mendel was able to figure this out without even, just by looking at the numbers, he was able to work backwards and figure out that there's a 50-50 chance that either allele is going to get passed on to your offspring. So it's pretty impressive. So because we know that genes are going to separate equally and separately, they we're going to be able to do numerous calculations to figure out probabilities because we know that the probability of getting either allele is going to be 50-50. And so we can use that idea then to set up these Punnett squares. So I'm guessing most of you are probably already pretty solid on this, but we'll do this one together, and then we're going to practice some more through the rest of the period. So this is our Punnett square. You might remember that we put the options for one parent on one side and the options for the other parent on the other side, right? So if this is dad here, he's heterozygous, he's going to pass on either the dominant or the recessive, 50-50, right? Half his offspring are going to carry one, half the other. Mom has the same genotype, so we're going to put her on this side. She's either going to pass on the dominant or the recessive, 50-50, right? So in these boxes, we're going to combine the allele that's above and to the left of it. So this box is going to end up having what genotype? Homozygous dominant. They're getting a dominant allele from both parents. This box is going to be heterozygous. This box is going to be heterozygous, and this box is going to be homozygous recessive. Okay. So we have how many different genotypes there? 1, 2, 3, right? This genotype is different from this genotype, which is different from this. Go ahead and you groups talk about what the phenotypes would be really quickly for each one of these boxes.
Purple, white.
All right, is that what you got? Okay. The other important thing with Punnett squares is that we have to be able to record the genotype and phenotype ratios. Is that ringing some bells for you? Okay, I do have a pretty specific way I want you to write those ratios, so we'll talk about that. So first, if we think about genotype, again, we have how many different genotypes here? Not 2 genotypes, 3 genotypes. Okay, 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive. But when you write the ratio out, I want you to use unreduced fractions.
Oh, so like 2/4.
Yeah. And I do want you to be specific. Don't just write the fractions out like what the genotype is. So for this, I would want you to put 1/4 homozygous dominant, 2/4 heterozygous, 1/4 homozygous recessive. That's going to make future calculations when we're dealing with multiple alleles easier, so that's why I want you to write it in that, practice writing it in that way. Questions on that? Okay. Phenotypes, we only have how many phenotypes? 2. So 75% are purple and 25% are white. If we write those ratios as a fraction, then it would be 3/4 purple to 1/4 white. All coming back to you now. Yeah. Yeah, this is hopefully pretty easy.
Yeah, it is so horny. You know, I love
Okay, we're going to do some more practice, but I'm going to introduce an idea here of something called a test cross. So I don't know if you necessarily need to take notes on this page, but we've already talked about how you can have organisms with the same phenotype but different phenotypes, right? So in this case, you could be homozygous dominant or heterozygous and end up both being purple. So if I have a pea plant with purple flowers, I can't tell just by looking at it which phenotype it has. What could I possibly do to figure out? Any ideas? Well, maybe it will give me a percentage chance, but we could do another generation. We would need to cross it with what? A homozygous recessive, yeah. So that is called a test cross. So if we have an individual, we don't know the genotype because it has the dominant trait. We can cross it, we can do another generation with a homozygous recessive individual. So again, that's called a test cross. So on the next slide, we're going to do 2 Punnett squares, one for each possibility. So if it's homozygous dominant crossed with homozygous recessive, what would the ratios be for the offspring? And if it was heterozygous versus that, what would the ratio of the offspring be? So take a couple seconds, try to set those up on your own. Yeah, just humor me and set up the actual squares. Yeah. So talk about those genotype and phenotype ratios with your group real quick, where those ratios going to be. Okay. So very big difference in the genotype and phenotype ratios here for these offspring, right? If our individual is homozygous dominant and we look at the offspring, all we're going to see is purple, right? So if we do this cross and all we get is offspring with the dominant trait, we know the individual was homozygous dominant. If we end up half and half, so half purple and half white, then we know that the individual had to have been heterozygous, right? So that's the point of the test cross. So if we look at the offspring from that cross, we can determine what the genotype of that parent was. Make sense?
Yeah.
Questions? So monohybrid Punnett squares, there's only so many combinations of genotypes you can do. Sorry, this is all animated in here. So only 6 possible combinations, really. So take some time to kind of work through those. You can set this square if you need to. If you don't need to set up the square, you don't have to. But see if you can list out the genotype and phenotype ratios for each of those crosses. Okay, so I would say this and this and this you should be able to do without thinking of Punnett square for sure. If you think about it. It's not the end of the world if you reduce them. I just all right, let's break for a hot second. Just talk about the 3 I circled. So if this is parent 1, it can only pass on what allele? The dominant, and this is parent 2, it can only pass on. So all the offspring are going to be homozygous dominant, and all 4 of them are going to show the trait that is dominant, right? Kind of similar. Parent 1 can only pass on the recessive. Parent 2 can only pass on the recessive. So all the offspring are going to be homozygous recessive, and all of them are going to show the recessive trait. This one down here, parent 1 can only pass on dominant, parent 2 can only pass on recessive. So all of those offspring are going to be heterozygous, and they're all going to show the dominant trait. If I look at this cross, this parent can only pass on the dominant. This parent could pass on either dominant or recessive. So there's two possible genotypes I could get, either T homozygous dominant or homozygous recessive. You can make the square if you need to, but it's going to be 50-50, right? Half and half. So 2/4. 2/4. Phenotype-wise, both of those genotypes are going to result in a dominant. So 4 out of 4 are going to show the dominant trait. This one's similar. Parent 1 has to pass on the recessive. Parent 2 is either going to pass on the dominant or the recessive. So half of the offspring are going to be heterozygous. Half of them are going to be homozygous recessive. If we translate that to phenotypes, half of them are going to be dominant, half of them are going to be recessive. And this is like the first Punnett square we did, right? So if both parents are heterozygous, we're going to get a ratio of 1:2 to 1. 1/4 homozygous dominant, 2/4. So these are kind of like the genetic multiplication tables. The more you look at these, the more you'll have them just memorized, and you won't have to make a Punnett square. But I am going to ask you to actually draw out the Punnett squares for your quick check, just to make sure we have that down pat. Questions on any of that, though? Okay. All right, so 6 questions on this. Half of them are about cat traits. There's a little key at the top about the cat traits. On the back, you're dealing with pea plants. There's a key at the top for those questions. I think you should definitely be able to finish before you leave. And similar to yesterday's quick check, if you can just take a picture of this, put it in the Google Doc, upload it to Canvas when you're done. Feel free to, I would maybe try to do it individually first, but if it's back with your group number.
Let's do the best, bro.